Let's have a little refresher about Mendel's laws. So Mendel, remember, he had his first law, said that when big A and little a, when sex cells are formed, big A and little a separate, right? You have segregation of alleles in the formation of sex cells. In his second law, it dealt with how do different genes transmit to sex cells together. And in that case, it said that big A is just as likely to go to the sex cell with big B as it is with little b. And that little a is just as likely to go with big B as with little b. And that's really kind of just the summary of what Mendel's laws are. But why? Why is it that they separate during sex cell formation? So we're going to talk about chromosomes some more now, because remember that every one of these genes has a physical location. Oops, well, that's a very ugly chromosome, but let's pretend that that's a chromosome. Oh, and here's the homolog. Oh, these are really bad chromosomes, but just to illustrate the point that they have a physical location on the chromosomes. So these would be autosomal chromosomes. Let's say it's chromosome number 3. And in this individual that's heterozygous, one chromosome would have the dominant allele and the other would have the recessive allele. They're alleles of the same gene, but they're different varieties, different flavors, or different alleles. So really the basis of Mendel's laws have to do with how these chromosomes are transmitted from parental cells to daughter cells. And so we're going to take a look at how chromosomes are transmitted or how they're inherited in cell division. So today's topic is going to start with the chromosomal basis, for Mendel's laws, or for the segregation of alleles, his first law, and independent assortment, the second law. So before we get into transmission or inheritance of the chromosomes and how they transmit, let's talk a little bit about chromosomal composition. So the number of unique chromosomes in an organism, in the cell of an organism, is the haploid number. So the haploid DNA content, the haploid chromosomal content, the haploid number is equal to the number of unique or different chromosomes in the nucleus of a cell. So how many are different? So in humans, the human haploid cell number, the number of different chromosomes in human cells, we call it the N number. In humans, there are 23 unique or distinct chromosomes. And so that's the haploid number. Now we have a haploid number of chromosomes in sex cells. So sex cells are haploid, meaning they have 23 total chromosomes. And of these 23, we know that 1 through 22 are the autosomes, and either X or Y. To make the 23rd. So there are 23 unique chromosomes. Sex cells are the only haploid cells in humans or in mammals. Some species can have part of their cell or part of their life cycle as a haploid stage. We're going to work with yeast in lab in the future, and they spend part of their life cycle as a haploid adult cell. In humans, in other mammals, in most animal species, the only time you see haploid cells is in the sex cells. most cells of mammals and of most animals are diploid. So most eukaryotic cells are diploid. In diploid organisms, have two complete sets of unique chromosomes. So they're 2N. They have two sets of those unique chromosomes. So human somatic cells are diploid. So our human somatic cells are 2N. And so that means they have two sets of 23 for 46 total chromosomes. It's two sets of 23. The depiction of visualization of the chromosomal composition in an organism is the karyotype. So it's a visual arrangement of the chromosomal composition. And chromosomes are passed on each time a cell divides. So chromosomal inheritance chromosomes are passed on with each cell division. So each time a cell divides, the chromosomes are passed on to the daughter cells. So let's talk about this process, that passing on of the chromosomes. And so we have to think about a cell, because whether we're talking about a somatic cell dividing by mitosis or the formation of sex cells by meiosis, all cells have sort of a life cycle, which we refer to as the cell cycle. Cell cycle. So it's the process of cell growth and division. Now, most adult cells, most cells that are like in our liver or neurons, most cells, once they get to their fully differentiated stage, really aren't in an active stage of dividing unless they get a particular signal to actively undergo cell growth and division. We call that sort of resting stage where cells are just sitting around doing what they're supposed to be doing, the GO. This is a non-dividing cell. So it's not actively undergoing growth and division. But cells, stem cells that you've probably heard of, particular types of body cells like your blood cells, especially if you have an infection, skin cells, intestinal cells, are actively undergoing growth and division. And we can break the cell cycle down into two main categories, and I'll just kind of give an inner overview of this first. So there's the inner phase, which is where the cells grow and where the DNA is replicated, followed by the division phase, which is mitosis in somatic cells. But in the formation of sex cells, you still have the interphase where the cells grow and the DNA is replicated. But that interphase is followed by a meiosis 1 and a meiosis 2. So two divisions. So in somatic cells, we have interphase followed by mitosis. In sex cells, we have an interphase followed by meiosis 1 and meiosis 2. So these are really mitosis and meiosis are where the chromosomes get divided up. These are sex cells or gametes. So interphase can be broken down into distinct substages. So the phases of interphase can be broken down into G1. In G1, the cell grows, so it actually increases in size. So the cell grows, it takes on size. Organelles begin to replicate. And a bunch of new proteins are made. So there's protein synthesis, and particularly proteins that are going to be involved in DNA replication. So those recipes in our genome that code for proteins that are involved in DNA replication, those start to really be expressed in G1. In G1, there's also a signal sort of needs to be sent to the cells to say, hey, let's progress now into the next phase. We won't really talk about that, but there's a G1 checkpoint. That has to be passed, but once this is passed, and like I said, maybe when we get into the cancer cell bio at the end of the semester, we'll come back to these. But once a signal is sent that says, okay, cell, it's time to divide, then it progresses from the G1 phase into the S phase, which is the synthesis phase. What is being synthesized? This is where DNA replication occurs. So all the DNA in the genome is replicated, and so you've doubled the DNA content by the end of the S phase. Following the S phase, once the DNA has all been replicated, then it enters into a G2. The G, by the way, stands for gap2. In the gap2, the cell continues to grow. Those organelles continue to replicate. And a new batch of proteins are synthesized, and these are proteins that are going to be involved in the actual division process. So more proteins are replicated. Not replicated, but proteins are synthesized that are going to be necessary for mitosis or meiosis. And then there's a G2 called a G2M checkpoint. And at this checkpoint, what happens is that the DNA is checked. This DNA that was replicated, it's checked to make sure that the content is correct. If the DNA did not replicate correctly, the cell undergoes cell death, otherwise known as apoptosis. If it does passes the checkpoint, then if it's a somatic cell, it's going to enter mitosis. And if it's a sex cell, it's going to enter meiosis. And this is really going to be our key one. The formation of the sex cells is the basis for inheritance in Mendel's laws. But we're going to look at the stages of mitosis first. Here is just to show you the haploid DNA content. So this would be haploid DNA or chromosomal composition. So this is what you would expect to see in a human sex cell, either an egg or a sperm. We have only one copy of each of the chromosomes, and there are 23 total. And the karyotype you're used to seeing, this is the karyotype of what you would expect to see for chromosomal composition in a somatic cell, the diploid content, where you've got 2 copies of each of the chromosomes. Here's a depiction of the cell cycle. Notice that the bulk of the cell cycle is spent in this preparation phase, the interphase. So you can think also of interphase as sort of preparation in DNA synthesis. Gap 1, the synthesis, and then gap 2. Once these sort of growth and replication phase is done, then we're going to move into mitosis, which is also going to be broken up into substages, but that is where those newly replicated chromosomes are going to get divided up. And so let's talk about the process of mitosis. So in the big picture, if I give sort of an overview, if we think about a somatic cell, before it goes into the cell cycle, it's got 2 copies of each of the chromosomes. After the S phase, all of those chromosomes have been doubled. So I actually like to think about it sort of like we've doubled our chromosomal composition. We've doubled our DNA content. We've doubled our DNA content. And then in mitosis, what happens? So the mitosis is that doubled DNA gets split up into 2 equal daughter cells. So this is sort of the big picture overview of the cell cycle of a somatic cell. After the S phase, we doubled their DNA content. After mitosis, by the end of mitosis, we have 2 identical daughter cells. We can call this the mother cell or the parent cell. And after mitosis, we have 2 identical diploid daughter cells. So let's look at the stages of mitosis. The first stage, and there's more detail in your book than I'm going to require you to know. I just want you to know what I'm going to teach you here. So the first phase is prophase. In prophase, the DNA is in a structure with proteins, remember, called chromatin, right? So in a resting cell, in a dividing cell, before it goes into mitosis, chromatin is how the DNA is packaged. That chromatin, which is kind of more loosely, you can think of it kind of as like a pot of spaghetti in the nucleus of a cell. The chromatin in prophase condenses into the structure that we commonly think of as chromosomes, and I'm going to call them dyad chromosomes. Those dyad chromosomes containing 2 identical sister chromatids. Held together at the centromere. So let me try to draw these. I probably should have practiced this. Here. Now think about this as prior to DNA replication. Even though it would not be condensed like this, it would be more like a long linear sort of thing like this. But if we visualize it just for purposes of handiness, just to be handy. So this would be like a pre-S phase. But we can kind of in our mind visualize this as a condensed structure. So before the S phase, you have an individual sort of unit of chromatin. After the S phase, you have replicated it. So what you have here is a chromosome. It's a chromosome with 2 identical sister chromatids. Held together at the centromere. So let's take an image of an electron, or take a look at an image of what one of these looks like. Here you can see an electron micrograph of a dyad chromosome held together at this constricted area that is the chromosome, I'm sorry, the centromere, and with 2 identical sister chromatids. This is a cartoon sort of representation of it, showing that there's these identical sister chromatids held together at the centromere. Now, this is just one of these pairs, right? So if we can think about this, we can go back here. In the cell, you would also have the homolog to this. And I'm just going to kind of put it over here, and I'll make it with little hash marks. So these are the same chromosome. Let's say it's chromosome number 3. This would also be the homologous chromosome number 3. But it is a separate chromosome. It is a separate chromosome. These are homologs to each other. These are sisters. These are sisters. These are non-sister homologs. So in prophase, the chromatin condenses. We have distinct units, these dyad chromosomes, which are 2 sister chromatids held together at the centromere. In the next phase, prometaphase. In prometaphase, the major event that's occurring in prometaphase is that the nuclear membrane dissolves. Because remember, DNA is in the nucleus of a cell. So these would be inside here. The nuclear membrane dissolves. And specialized microtubules called spindle fibers attach to protein structures at the centromere called kinetochores. So specialized microtubules called spindle fibers attach to specialized protein structures at the centromere called kinetochores. So the centromere is sort of a region. Kinetochore is a physical structure. I could maybe I can make some kinetochore looking structures. Let's say in blue here. So specialized microtubules called spindle fibers attached to the kinetochores. If we look at these actual images of cells that have been fluorescently stained, where the DNA is shown in blue, notice that in a cell prior to mitosis, that the DNA is diffuse. And if we look at the cartoon picture, it kind of looks like a pot of spaghetti. As in prophase, what happens is that these chromatin starts to condense into those distinct units that we think of as classical sort of centromeres. And then in prometaphase, you can see here that these cytoskeletal structures called spindle fibers attached to the centromere. And these spindle fibers are going to ratchet the chromosomes into the center of the sphere here in metaphase, right? So the process of metaphase, these chromosomes are going to line up at the cell's equator called the metaphase plate. So in our next stage, in metaphase of mitosis, the chromosomes line up. And I like to think of it as single file. So they're not in pairs, they're independent of each other, but they're lining up on this equator. So they line up at the equator of the cell called the metaphase plate. So if I kind of depict it here, I'm just going to make my chromosomes look like, let's make a big chromosome, let's make a little one, let's make another big one, let's make another little one. So notice that while these two might be homologs to each other and these two might be homologs to each other, they're not attached to each other at all. They're single file, and they're lining up at the equator of the cell or the metaphase plate. They got there due to those cytoskeletal structures called the spindle fibers, which sort of ratchet them to the center of the cell. So here's metaphase. Now there would be, in a human cell, there would be 46 of these guys lined up. There would be 46 of these guys lined up at the metaphase plate. In anaphase, these spindle fibers start to shorten. They're big polymers of these microtubule subunits, and they start to shorten. And as they shorten, you get the separation of these sister chromatids. So the spindle fibers shorten. And by doing so, they pull apart the sister chromatids. And the sister chromatids separate and are, or you can think of them, they're pulled towards opposite poles. Towards opposite poles of the cell. So if we zoom this forward in anaphase, what you would have is one batch of sisters in one area, and the other batch of sisters in the other. In a human cell, that would be a total of 46. Right? The final phase, and I'm going to just lump these two together, are telophase and cytokinesis. And that is where membrane invaginates and you get the formation of two distinct cells. The nuclear membrane is going to reform. So in here, the nuclear membrane is going to form around these sister chromatids, and you get the separation or the division into two distinct cells. So let's take a look here at some images. Here's anaphase. You can see these guys getting pulled apart. Ultimately, they're on opposite ends of the cells. The cell membrane is going to begin to invaginate, and this process is going to continue all the way through until it pinches off and you get two distinct daughter cells. And they will each have 46 chromosomes, just like the starting parent cell. Here's another little cartoon depiction of what's happening. And then I have an animation here of mitosis in a real cell. So the microtubules are stained in fluorescent green, and then in this case, the DNA is stained in red. And so let's take a look at a time-lapse video of a cell that's actually undergoing mitosis. You can see metaphase, they're lining up at the equator of the cell, and then the sisters get pulled apart, and they will decondense, and the new cells will form.